Professor Dr. Wan Faiziah to present her research on transcriptomic profiling of early onset estrogen receptor positive breast cancer. The one floor is yours.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. And good morning to everyone. Thank you, Madam Chairperson, Prof. Faiziah, for your kind introduction. Thank you to organizer for inviting CRO to present our work today. Salam Adil Fitri to all Muslim. Selamat Hari Raya Maaf Zahir Batin. Today, I would like to share our research work on transcriptomic profiling of early onset ER-positive breast cancer. So why this study matters? Breast cancer, as you all know, that breast cancer is the leading cancer among women worldwide. Interestingly, studies showed that breast cancer in Asia presents earlier, which is 10 years younger than in Western, and behave more aggressively. This raises the question, is early onset breast cancer biologically different? So that's why this research coming. And then why transcriptomic? I forgot to write here, why transcriptomic profiling? What is the transcriptomic? It comes from the word transcript, meaning the RNA molecules. So transcriptome means the complete set of RNA molecules produced by the particular cells. And then transcriptomic profile, meaning that the analysis of all the RNA transcript within cells or tissue at specific time to reveal the active gene expression pattern and providing insight into the cellular state and also the mechanism of the disease. Okay, because of this clinical dilemma, you know that breast cancer in young patients usually aggressive pathological features, high rate local regional recurrence. Because of this clinical dilemma, we hypothesize that the young breast cancer possess a unique, highly aggressive biological profile. Then we tested this assumption in ER-positive breast cancer, which account for over 70% of all breast cancers. We approach the study from clinical, molecular, and microenvironment perspective. For the clinical, we compare clinical pathological characteristics. For the molecular, we compare the transcriptomic profiles of young and older age, and also we assess the protein expression levels of proteins that encoded by the differential expressed genes in young and also in older women. And also we include the microenvironment in which we compare the tumor stroma ratio and also the tumor infiltrating lymphocyte levels. Right, briefly, the study was conducted in several components. First, clinical pathological analysis involving 570 breast cancer from HPUSM. All right, and then the second, the transcriptomic profiling. We analyzed 12 fresh breast cancer tissue using DNA microarray, followed by bioinformatic analysis using R software. And then the third validation study, where we combine our sample with TCGA data, the cancer genomic atlas data that totaling over 1,200 cases, and perform qPCR in silico analysis. And that's the protein expression was assessed using immunohistochemistry on 74 formalin-fixed paraffin embedded breast cancer tissues samples. And finally, the tumor microenvironment was evaluated in 205 breast cancer tissue. All right, and in which we assess the tumor stroma ratio and also the tumor infiltrating lymphocytes. All right. Okay. This slide outlines our study workflow, where we combine the clinical pathological data with tumor samples, followed by the RNA extraction and transcriptomic profiling. Differential gene analysis and pathway evaluation were performed, and key genes were validated using the RT-qPCR and immunohistochemistry through statistical analysis. For better understanding from the workflow, there are three focus investigative pipeline. Okay, first we perform transcriptomic discovery using DNA microarray to identify the key differentially expressed genes between young and older patients. And the second, the discovered differential expressed genes were then strengthened through in silico validation using the large external data set like the Cancer Genomic Atlas from the free website. And then the third, we translated these molecular signals into real clinical relevance by confirming protein expression using IHC in an independent cohort. And this ensures our findings are not just statistical, but biologically meaningful. All right, and then having established our pipeline and validation strategy, we now move to the result. We first examine the association between age and clinical pathological features. And you can see, as what you can see here, notably younger patients were significantly associated with larger tumor size, particularly tumor that are greater than 5 centimeter, and a higher proportion of luminal B subtypes. We also observe trend toward more bilateral breast cancer in younger patients. And overall, these findings suggest that younger age is linked to more aggressive disease presentation at diagnosis. We then identify significant transcriptomic differences expressed in young patients from over 21,000 genes analyzed. Only 12 show significant differential expression. Among these, 8 genes were upregulated in younger patients, mainly associated with metastases, proliferation, and extracellular matrix modulation. Conversely, 4 genes were downregulated, which are linked to tumor suppression, hormonal response, and also cellular stability. This suggests that early onset breast cancer has a distinct, potentially more aggressive molecular signatures. Pathway enrichment analysis revealed that these upregulated genes are significantly involved in focal adhesion and extracellular matrix receptor interaction pathway. And these pathways are critical for tumor cell adhesion, migration, and also invasion. And these findings support the idea that tumors in younger patients may have enhanced metastatic potential driven by tumor-stroma interaction. To validate our findings, we perform in silico analysis using the Cancer Genomic Atlas data. And out of the identified genes, several key signatures were consistently confirmed, and upregulated genes such as ACVR2A and SERPINE2 were associated with tumor growth and metastases, while the downregulated genes, including the RAN, BP3L, GAIA-TIL, and ESR1, were linked to increased migration, poor prognosis, and potential endocrine resistance. And this reinforced the robustness and the clinical relevance of our transcriptomic findings. Survivor analysis demonstrated that loss of down-regulated genes, particularly the RAN, BP3L, GAIA-TIL, ESR1, SFX, and 2, was associated with significantly shorter relapse-free survival. In contrast, higher expression of selected upregulated genes correlated with better outcomes. And this indicates that these genes are not only biologically relevant, but also carry prognostic significance. We further validated gene expression experimentally using RT-qPCR. Our results confirm that both RAN-BP3L and also gliatil-1 are significantly downregulated in younger patients compared to older. And this provides a direct biological validation of our transcriptomic data. Importantly, gene expression doesn't always reflect protein levels. All right. Therefore, we extended our analysis to protein expression using IHC in a larger cohort, and our aim to determine whether the observed gene downregulation translates into actual protein loss, which would strengthen its biological and also clinical relevance. We next evaluated protein expression of RAN-BP3L using immunohistochemistry in 74 breast carcinoma cases. RAN-BP3L shows strong nuclear expression in normal breast ducts. Okay, but the signal progressively diminished in higher histopathological grades. Consistent with this, the higher RAN-BP3L expression is significantly associated with lower grade tumors, while no significant association is observed with a patient age. Functionally, loss of RAN-BP3L may contribute to tumor progression through activation of the BMP signaling pathway, leading to accumulation of the SMAD4 proteins and upregulation of genes involved in extracellular matrix remodeling and metastasis. Together, these findings support RAN-BP3L as a potential tumor suppressor that link to disease aggressiveness rather than age. We next evaluated gliatil-1 protein expression using IHC in 56 breast carcinoma cases. Gliatil-1 shows strong cytoplasmic expression in invasive carcinoma. Importantly, loss of the gliatil-1 is significantly associated with positive lymph node status, indicating a link with more aggressive disease. However, consistent with our overall finding, there is no significant association between gliatil-1 expression and with a patient age. Gliatil-1 function as key detoxification enzyme under the normal physiological condition. It catalyzes the conjugation of carboxylic acid derivative with amino acid, such as glycine and glutamine, facilitating solubility and efficient excretion of metabolic product. However, in breast cancer, the loss of gliatil disrupt this detoxification pathway, and this lead to the accumulation of harmful metabolite, dysregulation of cellular energy metabolism, and activation of stress-related signaling pathways. Ultimately, these changes contribute to loss of cellular hemostasis and promote tumor progression. We next evaluated the tumor microenvironment features, specifically the tumor stroma ratio and tumor infiltrating lymphocytes, TIL and TSR. Okay, for TSR, a lower TSR, meaning that lower stroma, indicating less stroma component, was significantly associated with older patients in our study that suggests a more favorable tumor biology in this group as compared to young. In contrast, analysis of tumor infiltrating lymphocytes, TIL, showed that higher TIL levels were significantly associated with post-chemotherapy cases that reflecting treatment-related immune activation. However, importantly, TIL levels showed no significant association with patient age. Overall, these findings further support that tumor microenvironment characteristics are influenced by tumor biology and treatment status rather than age itself. Traditionally, we believe that young age itself drives a unique transcriptomic profile leading to more aggressive breast cancer. However, our data suggests a paradigm shift. It is not age directly driving these molecular changes. Instead, the younger patients tend to present with more aggressive tumor features, such as higher grade of tumor and lymph node involvement. And these features are what shape the transcriptomic profile. In essence, the genes respond to tumor biology, not the chronological age. In the raw data, we initially observed 12 differentially expressed genes, DEG, between young and older patients. However, once we adjust for clinical severity, specifically the tumor grade and nodal status, these differences completely disappear. And this demonstrates that the observed transcriptomic differences are not driven by age itself, but by the underlying tumor biology. So our findings highlight three key clinical implications. First, at the transcriptomic level, ER-positive breast cancer appears biologically homogeneous across age groups. And the second, the true drivers of molecular variation are clinico-pathological factors, particularly the tumor grade and also the lymph node status, not the chronological age. And finally, this has important clinical implications. Treatment decision, risk stratification, and targeted therapies should be guided by tumor biology rather than age alone. And in essence, we should move away from age-based assumptions and focus on what truly defines the disease. To conclude, from a clinical perspective, early onset ER-positive breast cancer present with more aggressive features, larger tumor, higher luminal B subtype, and increased bilaterality. At the molecular level, we identified the key transcriptomic alteration, including the downregulation of RANBP3L and gliatil-1 that are associated with pathways such as SMAD signaling and BMP activation and metabolic stress that supporting the tumor progression. And from the tumor microenvironment perspective, features such as tumor-stroma ratio and tumor-infiltrating lymphocytes were influenced by tumor characteristic and treatment status rather than age itself. Importantly, integrating all three perspectives, our findings consistently demonstrate that these differences are driven by tumor biology and disease severity, not the chronological age. And this support a shift toward biology-driven rather than age-based clinical decision-making. Our heartfelt thank you to those who involved in this research, especially to my PhD student that graduated on time in 2023, Dr. Ala Siddiq, and to our co-supervisory team, AP Dr. Sarina, Dr. Maya Mazwin, Dr. Ahmad Demetri, Dr. Siti Narsikin, and our collaborator, AP Dr. Razaimi Razali from Kota University and Prof. Efrat Lam from Griffith University. And to PPSP, Pathology Department, Human Genome Center, Department of Surgery, Nuclear Oncology Department, and RNI, HPUSM to Bestari team, thank you so much. And thank you to Ministry of Higher Education for funding the FRGS grant. And we are grateful that this work has been translated into 3 publications, marking an important step in contributing to the evolving landscape of breast cancer research. And Selamat Hari Raya again, Maaf Batin from my family and me. And with that, I thank you.
Okay, thank you, Prof. Faiziah, for enlightening presentation. So now we have quite time. We have another 9 minutes. So I would like to invite more questions from the audience and also especially from our judges. You can post the question live. I think that will be more stimulating for the session.
Hi, morning. Chan's here.
Yes, Prof. Chan, we can hear you.
Yeah, okay, thanks. Prof. Thanks for sharing with us all these very exciting information. But I believe that you already have some plan for your further study based on your result, right? Do you mind to share with us what is your further plan about your research based on what finding that you have found?
Thank you, Prof. Chan. Thank you for that question. Yeah, expected question, but still, I have no idea how to answer. But yes, of course, because we have the key genes with for the younger patient, especially for the RAMBP3L and the gliatil-1, and we know that that is very significant expressed. And of course, I would like to proceed with a functional analysis for that genes. And of course, one of the limitations in our study, we found that the sample size, because the sample is equal to the cost. Yeah, so we like to, if I plan to apply for another grant to proceed with the study and with a larger sample. At this time, maybe not using the macro RNA, maybe to another method, RNA sequencing, possibly, and maybe to integrate, to add a sample by using the multi-center study as well. Thank you, Prof. Chan. That's my plan and also my planning and also maybe my dreams.
Okay, thank you.
Okay, we can entertain more questions from the audience or from the judges. Again, from the judges.
Good morning.
Good morning. Yes, we can hear you.
C2 from PPSK.
All right. Yes, we can hear you.
Since we still have time. So it seems like the genes you identified to be differentially expressed, either overexpressed or you hear.
Yes, we can hear you.
Right. So the differentially expressed gene, it seems like it's not age dependent, but more tumor biology dependent. So in your future study, are you going to focus more on tumor biology instead of age?
Thank you, thank you, Prof. Situ. Yeah, but because my interest is still in working on the young breast cancer.
Yeah, that's why I asked.
Yeah, maybe I need to get your opinion as well, whether because from this study we found that it's not direct correlation with the age, chronological age, but more on that aggressive biology behavior. Yes, I think that's a good question, a valid question. Maybe we not focus on age anymore, but we still from the study we still found that that genes still expressed in younger, and that young patient correlate with the aggressive behavior, something that. Maybe I still aim for the young age with more sample size to proceed with the further functional analysis and further study.
I understand the feeling because I thought the main theme is to differentiate young and old. But unfortunately, the results did not manage to help you to differentiate. That's why I asked, and it seems like you still want to pursue a differential express gene in young and old patients. Yeah. Okay, thank you.
All right, thank you, Prof. Situ.
So thank you to both judges for the questions. I think we 9:07. I think it's time to close the session. And before I close the session, I would like once again to thanks to both our presenters, Dr. Wan Fatihah, and also Prof. Wan Faiziah. And also thanks goes to our audience. Despite we are still in the holiday mode, I can see we have 100 plus participants for today's session. So thank you everyone, and Assalamualaikum warahmatullahi wabarakatuh.